Hello everyone, welcome to ITLS Academy. I am your mentor Arnav, and today we are going to discuss about the topic basics of DNA replication. So at first we should know what is DNA replication. DNA replication is a biological process through which two identical replica of DNA molecule is produced from one parental DNA molecule. This process is found on all living organisms and it is the most essential part for biological inheritance. Now we will learn that in which process DNA replication occurs. There are three assumptions. Those are conservative method of replication, semi-conservative method of replication and dispersive method of replication. We will learn about that now. Now see, in case of conservative mode of replication, it is the parental DNA molecule and after first round of replication, two new DNA are produced. Among the two new DNAs, one is the copy of that parental molecule and one is the newly synthesized daughter strand. So, in this type of method, the parental strand is fully conserved. That's why it is known as the conservative method of DNA replication. Now see, in case of semi-conservative mode, look, here is the parental strand and in the first round of replication, one strand from the parental copy and one strand from the daughter copy is forming the new DNA. So in this case, the parental DNA molecule is not fully conserved, so it is known as semi-conservative. And in case of dispersive mode of replication, see, here is the parental strand and newly synthesized DNA molecule is a combination of different fragments from the parental strand and the daughter strand. Now, in which process DNA replication occurs? That was a big question, and that question was answered by the experiment of Michelson and Stahl. In their experiment, at first they grew the cells of E. coli in N15 medium. After that, they grew those cells of E. coli in N14 medium. Okay. Now, N15 is the heavy isotope of nitrogen and N14 is the light isotope. So, what the result they got after watching the replication of E. coli cells? Look here. So, here in the generation naught, it was N15 and N15, both the DNA strand. In the generation 1, it was a hybrid N15 and N14. And in generation 2, it was found that it was N15, N14 and N14 and N14. So from this result, what we can conclude? We can conclude that DNA replication is done in a semi-conservative way. If DNA replication was done in a conservative way, then after the generation naught in generation 1, two N15 strand would have formed and one N14 strand would have formed. But in this case, they got the hybrid of N15 and N14. And if DNA replication was done in a dispersive way, then in the generation 2 they would have found both the hybrid, but they found one hybrid and one N14 and N14. So from the experiment of Michelson and Stahl, we can conclude that DNA replication is done in a semi-conservative way. Now we will discuss about the general feature of DNA replication. First point is DNA replication is semi-conservative. We have discussed about it in the earlier slide. Second point is it proceeds from a specific point called origin of replication. That means DNA replication is initiated at a particular site of the DNA and that site is known as origin of replication. Third point is it proceeds in 5' to 3' direction. That means whenever a new daughter strand is synthesizing, its direction is 5' to 3'. Fourth point is it occurs with high degree of fidelity. Fifth point is it is a multi-enzymatic process. That means more than one enzyme is involved in the process of DNA replication. Now in this slide we will discuss about the different stages of DNA replication. There is three stages of DNA replication: initiation, elongation and termination. In the initiation phase, the double helix of DNA is unwinded and RNA primer is synthesized. The complex is known as primosome. In the elongation, the DNTPs are added in the daughter nucleotide and the complex is known as replisome, and the termination of the DNA replication is done by a particular protein, and that protein is known as TUS protein or TUS protein, and termination occurs at a particular site, and that site is known as TAR termination site. Now in this slide we will discuss about the basic requirement for initiation of DNA replication. Now see here, that is the double stranded DNA. It is the double stranded DNA, and that DNA is need to be unwinded, and the work of unwinding is done by the enzyme helicase. Okay. Now after the two strand has been unwinded, the protein ssb means single stranded binding protein, binds with the unwinded strand, and it ensures that it doesn't form the double helix again. Third one is topoisomerase. Whenever helicase is uncoiling or unwinding the double stranded DNA, there is a pressure created at the DNA molecule, and that create supercoil, and that supercoil is removed by the enzyme topoisomerase. Fourth one is primase. The enzyme DNA polymerase requires a 3' OH free primer. That primer is RNA primer, and that is synthesized by the enzyme primase. Now in this slide we will discuss about the different type of DNA polymerase enzymes found in prokaryote. These are DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. There are two additional polymerase which are found on the prokaryote. Those are DNA polymerase 4 and DNA polymerase 5. Now in this slide we will discuss about the function of different DNA polymerase enzyme. DNA polymerase 1, it help in repair and it also help in primer removal. DNA polymerase 2, it mainly help in the DNA repair, and DNA polymerase 3 is the enzyme with high processability, so it is the major enzyme for chain elongation or polymerization. Now in this slide we will discuss about the structure of DNA polymerase 1. We all know that an enzyme is a protein. So a protein has a N terminal and a C terminal. If we clip DNA polymerase 1 with a protease enzyme, then it will yield a small N terminal fragment and a large C terminal fragment. The large C terminal fragment has 5' to 3' polymerase activity and 3' to 5' exonuclease activity, but it lacks 5' to 3' exonuclease activity. The N-terminal part has the 5' to 3' exonuclease activity, and this activity is only found on DNA polymerase 1. Now in this slide we will discuss about the structure of DNA polymerase 3, which is the major enzyme involved in the process of replication and polymerization. Now see, from the diagram we can assume that the DNA polymerase 3 is a multi-subunit structure. Among different subunits, this function as a catalytic core. The beta subunits form the sliding clamp, and the sliding clamp is loaded on the replication site by the gamma subunit, and these two multi subunits are attached together by two tau subunits. So it is clear. So thank you everyone for listening to our lecture. If you have any doubts regarding this video lecture, you can ask your queries in my mail address or you can text me on this WhatsApp account. So thank you everyone.